Well, uh, yes, just welcome to the second panel of the fourth International Online Symposium on Meteria Medica with the topic of Fenugreek. We enjoyed listening to the lectures of the speakers in the first panel regarding the applications and therapeutic uses of Fenugreek in different traditional medical systems. And now we're starting the second panel with a focus on some recent studies on somehow modern aspects of fenugreek. As the first speaker of this panel, I would like to invite Dr. Dilip Ghosh. Dr. Ghosh is the director of NutriConnect in Sydney, Australia. And we are also honored to have Dr. Ghosh as the visiting professor at our school, School of Persian Medicine in Tehran University of Medical Sciences. So, dear Dr. Ghosh, it's a very great honor for us to have you here today as our speaker, and thank you for accepting our invitation. The platform is yours.
Thank you, Professor Rudabe, and welcome everybody on the board. So I am trying to share the screen. So can you see my screen?
Yes, it's visible. Thank you.
Okay, thank you very much. So, you know, most of you here, I think you are very well expert in this area, Fenugreek, and I believe you have good knowledge in modern nutraceutical industry also, how the product is developed and that sort of things. And definitely you are expert in traditional medicine, complementary medicine, Ayurveda, traditional Chinese medicine also. So today I am going to discuss the modern aspect of fenugreek, that is the evidence-based nutraceutical properties and therapeutic potential. So first, the disclaimer. I tried to uh, gather uh, most up-to-date information on this area, and I try to made 100% accuracy also. And I just declare if I mentioned any brand or any particular research. So I have no financial connection with any of this company, so named in this presentation. This is just for the sake of uh, science and your understanding. Just as I mentioned, most of you are in this area, so you can, when you have time, go through these all these books I edited or I wrote in last 7-8 years. So these are very exciting area, uh, clinical aspect, biotechnology, commercialization, how the entire pharmaceutical or sick industry, pharmaceutical industry moving to the nutraceutical industry. And the natural medicine also, I showed entire clinical efficacy, safety, and quality. And this is the brain health and beyond, how nutraceutical can help in this area. This one I published 1.5 years before. I think this is the most authentic book so far on fenugreek because we covered the traditional medicine to the modern medicinal use, and we exemplified few product or few science. So these are registered in the Western world as a medicine also. This one published last year, Nutrition Science, Marketing, Health Claims and Public Policy by Academic Press. So this one in the press, this is a very informative book, traditional, complementary and integrative medicine for neurocognition. Because the Tehran University Medical Science, although they are specialists in Persian medicine, but they are doing a lot of work on the integrative medicine. So that means uh, to compete in this area, especially for the pharmaceutical industry, you have to develop a model uh, to get all good things from all parts, like Ayurveda, traditional Chinese medicine, Persian medicine, even the pharmaceutical industry also. So we are targeting neurocognition as a first target. This one in progress. We are almost 80% done. Ashwagandha, the potential drug candidate for the ancient Ayurvedic remedy. As you know, ashwagandha is very hot in recent years. Hot means from commercial perspective, and also there are a lot of debate and controversies also. Okay, you all know what is food. It is a protein, carbohydrate, and fat used in the body organism to sustain growth, repair, vital process. Functional food. A functional food is a food claimed to have an additional function by adding new ingredients. That means it is not a food, but you are adding some new ingredients that make functional. Nutraceutical, a food or specific component of a food that allegedly provide medicinal or health benefit, including prevention and treatment. So this is the first time we are talking about health benefit and also the prevention and treatment of the disease. And the latest one, that is the medical food. Maybe you are aware of that. Today I am not going to discuss because I have only 30 minutes time. So medical foods are foods that are specially formulated and intended for the dietary management of a disease that has distinctive nutritional need, like Alzheimer patient. So they need some particular nutritional supplement or nutritional support. Normal food cannot provide these things. So you need to develop a new formulated food for this particular Alzheimer patient. So it can be given by the doctor. You cannot go and buy from the grocery shop or any other shop also. So it is a doctor prescribed food. This is called medical food. This is the definition of nutraceutical: medicinal and health benefit, including prevention and or treatment of a disease. So go to the subject. Fenugreek, this is the book. You haven't, many of these organizer, the Tehran University, they contributed chapter in this book also. So if you are interested, you can go through this. So we all know fenugreek. I'm not going to repeat. It's a leguminous plant found in India, Egypt, Middle Eastern countries, traditionally used as a flavoring agent, culinary spice, food. It is a text in the Ayurveda, Greek, Latin, Pharmacopoeia, USP, herbal medicine, and many more. Abundantly available. It is not a rare or shortly available herbs also. Generally recognized as safe. That is a grass in USA. So if anything has 100% safe to take inside, that is called generally recognized as safe. And daily safe limit is 100 to 150 gram. So that means if anybody take 100 grams or 150 grams of fenugreek, they won't feel any toxic or safety issue. So I worked with one leading Indian biopharmaceutical company. They have done huge study in the fenugreek from process development, extraction process, to preclinical trial, to the human trial also. If you see, this is the ideal scenario. Raw material that has to be DNA barcoding, on-site vendor validation, and QC tested. In process manufacturing, it should be a HACCP, regular audit, and manufacturing in a GMP environment. Finished product, rigorous testing of final product, and also not only the local heavy metal, but you have to comply with the California Prop 65. So this is the highest level of heavy metal testing compliance. USP standard, purity tested through the sophisticated HPLC and in-house stability studies. When you come to the efficacy, there are preclinical efficacy, safety, efficacy and safety in the human, and then you can go to the market. Extensive in vitro ex vivo studies, that is the animal studies, efficacy using unique and specific unique model, extensive data for the safety and toxicology like acute oral toxicity, 90 days subchronic, mutagenicity, chromosomal aberration, prenatal development, and the clinical study. Preferably, if you can do the double-blinded placebo-controlled trial with a good number of population, that would be the ideal. Then you can go to the market. So traditionally, we know people are taking the entire fenugreek seed, but here I am giving you some few example or new insight. So we isolated 4 component from fenugreek. One is the amino acid component that developed for glucose control or anti-hyperglycemic condition. Alkaloid, that is for antioxidant and protective. Glycoside, anti-inflammatory. Fibers, muscle building, fat reducing, lactation, and constipation. If you see that that my partner company, they have 88 patents only on fenugreek. Can you imagine how much committed this company for the fenugreek? And also total 101 publication, including clinicals, mechanism, and preclinicals. 88 patents, global patents, and 101 total publication only on fenugreek. Just giving you a snapshot, the amino acid, mainly the 4-hydroxy isoleucine, we isolated this part and we developed for the sugar control or diabetes control, mood, depression also. The green light means product already in the market. Black, we have done a lot of studies, but the product not in the market. Alkaloid, that is the trigonelline, that is mainly for the Parkinson disease or any neurodegenerative disease, that is organ protective, antioxidant, again mitochondrial oxidative stress. Glycoside, that is the testosterone booster, that is the free testosterone booster, post-exercise recovery, bone and joint health. Saponins for male libido increase and testosterone booster. Soluble fiber, mostly the glycomanans, that is body composition, lipid and fat management, that is weight management, body glucose control, and also blood lipid management. Insoluble fiber, digestive health, prebiotic, and modify food texture. Protein and fiber, when you mix, anti-hyperlipidemic, improve glucose tolerance, and reduced weight gain. So there are many areas you can develop product based on these 4 ingredients. That is the, you can see how much clinical trials have been published in this area. Saponin, 1, 2, 3, 4. Glycoside, 1, 2, that is the big study. Trigonelline, that is the alkaloid, that is 1. Amino acid, this is the lead publication. Soluble fiber, there is 2, and saponin, there are 2 more in this area. That is when saponin plus the glycoside also. So I am giving you 3 example. Forget about the product name. I am just giving you just to get you informed. That is called andropic, glycoside part of the fenugreek seed. We isolated the glycoside part and we have done the entire study from in vitro to animal study to preclinical to clinical trial. Proven efficacy and safety in 2 independent human clinical trials. Significantly increase free and bioavailable testosterone. As you know, we need free testosterone, not the total testosterone. When you are aging, like the 40, 45 plus, your testosterone level is going down. It is not only in male, also in the female also. So that means you have to increase the free testosterone level in your body, in your bloodstream. And that this glycoside part of fenugreek does this uh, for the human. Adulteration free and approved by the many countries like TGA in Australia, Health Canada, and Food Safety Standard Authority of India also. So that is well established, lot of studies, and also if I go through a quick two study one, single dose, that is a 600 milligram, 7 days, significantly increase androgenic hormone. You can see the andropic, significantly increase free testosterone, significantly increase total testosterone, and not only free testosterone, that has to be absorbed in your body. So that means bioavailable testosterone should be also increased. Uh, in this study, all these three parameters are very good. Study 2, a 2-week study in the USA. That is also a double-blinded placebo-controlled trial, 500 milligrams. Here you can see the bioavailable testosterone increase, total testosterone increased, and the additionally we show that is the body fat that is decreased. That means that is good for not only for the testosterone or but the body fat also decrease significantly. The second product is Sugar Heal. That means sugar healing, effective add-on therapy for the blood sugar management. When we call the add-on therapy, that means we are not encouraging patient to use only this for blood control, but it can reduce the, like if you are taking insulin, so that can reduce the dose of insulin, but it has to be used along with the normal anti diabetic drug. Sugar Hill is an innovative ingredient that has been clinically shown by the two clinical trials that significantly reduce the HbA1c of the type 2 diabetes. What is Sugar Hill? It is the 4-hydroxy-isoleucine, that is the amino acid, plus trigonelline, that is the alkaloid part. So, two published clinical studies supported healthy blood sugar level, maintain HbA1c. You can see the study 1. Here is the study 1. You can see 0 week 12, and this is the placebo. You can see there is a significant reduction of 155 to 146, 225 to 205, 7.4 to 6.88, and also the number of insulin doses required, that is consistently reduced. So if you are taking 4 doses of insulin every day, so maybe after 3 months you can reduce go to the 2 insulin dose also. So this is a very, very significant advantage. Fenugreek, this is the new invention. It is a fantastic invention used in the fenugreek protein and fiber. Fenugreek is debitterized. You know, the problem of fenugreek is very bitter. You cannot take straight. So using our patented technology, we debitterized, we defat also, because fenugreek has a lot of fat. So we remove that fat, we remove the bitterness, and it is a high, rich in fiber, high in protein, and has a added benefit of being ultra low in the carbohydrate. So that means it can manage a blood sugar level, lower glycemic index of high GI food, and improve gut health also. As you know, protein is a crazy thing in today's world, but this is a new protein people can use for gym or any other area with added benefit also. You can see the here protein not less than 25%. That means a very, very good amount of protein. Soluble fiber not less than 25%. insoluble fiber not less than 25%. So there is a good balance between soluble fiber and insoluble fiber, 25% protein, only 1% carbohydrate. So that means carbohydrate is very, very low. And you can see there are good amount of uh, amino acid: leucine, valine, isoleucine, tryptophan, proline, alanine, serine, glutamic acid, and aspartic acid. So if you can use as a cereal, you can use in a beverage, breads, noodles, powder, enhance the nutrient profile of the food. If you add this Fenugreek, that is the high fiber, high protein, low carb, low fat. And what are the benefit? You can use for diabetic patient, blood sugar management, lower the GI of the food, and improve the gut health. So there are good studies, human clinical trial also. So 2 studies have been done. Significant improvement is glucose variability, significant reduction in the fasting blood glucose level, and significant reduction is the in HbA1c. And in the another study, we showed significant lowering of the glycemic index also. Stimulation effect of the acetate, propionate, and butyrate, less gas production, because as I said, it improves your gut health also. So less gas production and bloating, and enrichment of the beneficial gut microbiota. So because it's a huge 25% insoluble and 25% soluble fiber, it enhances the beneficial gut microbiota also. Okay, so Katsi, I think Indus Biotech for the photos and the images. I acknowledge their help also. So I finish. If you have any question, I am happy to answer.
Thank you very much, Dr. Bush. Thank you very much, Dr. Bush. It was really interesting, and I think it was a bench to bedside approach. You used the results of the research, and you performed some research yourself, and then turned it into some products which can be used by patients at bedside. So it was a real bench to bench to bedside approach, and I'm especially interested in that Fenugreek. It sounds a very interesting product, and is something to be easily used by all those who need to somehow optimize their blood sugar and lipid profile. I think such sorts of enriched foods are really helpful in today's lives, because one of the big problems of patients is taking too many capsules and tablets, and if we can provide something therapeutic in the form of a food instead of a medicine, it would also increase the adherence and quality of life of patients. So thank you very much. And there's a question in the chat box. Okay, one of the audience is asking if Fenugreek can be used as prebiotics for the gut microbiome. Does Fenugreek have prebiotics properties for the gut microbiome?
Yeah, I think she asked about gut probiotics. So fiber is a prebiotic, as you mentioned. Yes, because it has a profound effect, very beneficial effect on the gut microbiota. And as you know, the ideal prebiotic should be balanced between insoluble fiber and soluble fiber. So that means Fenugreek, Fenugreek particularly, it has a beautiful balance of the soluble and insoluble fiber. Yes, the short answer is yes, you can use as a prebiotic also, because it helps the gut health also.
Thank you. And there's also another question asking about the somehow effect of Fenugreek on testosterone in female. Yes. The hormonal effect seems to be a bit confusing for our audience. Does it cause any problem of testosterone level if females take it?
Yes, I think it's a very good question. Uh, I think in general consumer mind, testosterone is a male hormone. Definitely, it's a that's why it is our product name is andropic. Andro means male hormone, and peak means it's going high. But as you know, uh, women also need testosterone uh, throughout the life cycle to keep their sexual or healthy lifestyle also. So, because I have only 30 minutes time, I couldn't get time to explain. We developed one product, we means with our Indian partner. Then they sell this product, the ingredient, to one big American company, and they are selling two products based on the similar ingredients. One is testofen, that is for male, and another is libifem, that is for female. So both different, slightly different structure, slightly different dose also, concentration, but purpose is same. Uh, that increase the free testosterone level both in male and female, and also it is not going very high, it is within the physiological level. So that means what you need you can get from this one, and it is only increase free testosterone, and it is not under the doping also. uh, you know this in Olympic or any sports, athletes are taking a lot of different things to increase the testosterone level, but this one, it is not a steroid or testosterone enhancer. It stimulates your body physiological process that remove or that release free testosterone from the bound testosterone. So that means it is not falling under the doping protocol also. That is the big advantage.
Thank you very much. And there's also another question about the safe daily dose of fenugreek for diabetic patients and for the case of dysmenorrhea. I personally think it highly depends on the type of the product and which component to be enriched on that specific fenugreek product. But would you please also share your experience, which daily dose is recommended for diabetes and dysmenorrhea?
Yep. If you see this, as I said, the amino acid fraction, especially the 4-hydroxyleucine, that is mainly developed for the diabetes control or blood sugar level. And they launch, the Indian company, they launch one product that is called Sugar Hill in India. That is not only Fenugreek, but 80% component Fenugreek with some supporting other Ayurvedic ingredients. So that means 450 milligram, that is the Fenugreek part, and some other synergistic. You know that sometimes you need some synergistic action that will synergize the effect of blood glucose level. But 4-hydroxyleucine is the main component from Fenugreek, that is the amino acid that helps controlling the blood glucose level. But personally, if you ask me, I cannot advise any advanced type 2 diabetic patient to take only Fenugreek to control their blood sugar. What your doctor is recommending, like metformin or injection also, so you can reduce the dose, significantly reduce the dose with the Fenugreek consumption. So that is the major advantage. That is the not a stand-alone treatment, but it is a adjunctive or add-on treatment.
Thank you very much. Yeah, that was a really important point to consider, that fenugreek should be used, or any other supplement for controlling blood sugar should be used under the supervision of a physician, because the dose should be adjusted along with other anti-hyperglycemic medicines.
Yes.
So thank you very much again, dear Dr. Ghosh, for sharing your experience and knowledge today with us. And let's go to the next speaker. I would like to actually introduce Dr. Andre Mesquita Marcus, PhD of Phytochemistry and lecturer at the Department of Natural Products, Osvaldo Cruz Foundation, Laboratory of Technology for Biodiversity in Health, from Rio de Janeiro in Brazil. Because actually Rio is 6 hours ahead of Tehran local time. It is very early in the morning, around 5 and 6 p.m. So it was not possible for Dr. Marcus to deliver his lecture online. He kindly recorded his lecture, and we are now listening to his speech, who's going to talk about the phytochemistry of fenugreek.
Salam. So, Bahir. My name is Andre Marcus. I work as a researcher in Natural Products Lab, TechBio, at Institute of Pharmaceutical Technology, Fiocruz Foundation, Brazil. It's an honor to be here and participate of the 4th International Online Symposium on Meteria Medica. So today we're gonna talk about exploring Fenugreek secondary metabolites. As you know, the species Trigonella phenugrecum, popularly known as Fenugreek, is used since ancient times in various parts of the world, mainly in Asia and Middle Eastern countries, where it has a great value in traditional medicine and used in food as a spice. So major Fenugreek producing countries are Afghanistan, Pakistan, India, Iran, Nepal, Bangladesh, Argentina, Egypt, France, Spain, Turkey, and Morocco. But today, the current largest producer of fenugreek species is considered India. The parts used are the seeds and the leaves. So in this picture, you can see the current location of the fenugreek producers all over the world. So as I told you, the main producers are the countries of Asia. Here is the South Asia, the Middle East countries, Egypt, Iran, and also the cultivation of fenugreek had spread over the Europe countries, especially France, Spain, and also Greece. In the last decades, also some sports have been appear in North America, in United States, and also in some countries of Africa. In the last years, has increased the number of clinical trials around the world. Here are the top countries that are investigating the therapeutical properties of fenugreek. So the top countries are United States, India, Iran, and also Australia. And the top topic research are related as diabetes or blood glucose, cardiovascular health, sexual function or hormone, and lactation. Talking about nutrition information, we know that one of the most parts consumed of this plant are the seeds. So primary metabolites are compounds essential for the plant growth, development, reproduce, and also have some particular characteristics. They are a source of energy and make part of the structure composition of the plant. So they are vital for the plant survival. They participate of several important reactions of catabolism and anabolism pathways. They are present in all plants in the world, and they are composed by carbohydrates, lipids, proteins, nucleic acids, and chlorophylls. So here is just a resume of the nutrition information. Almost half part of the content present in the seeds are composed by carbohydrates, 20 to 30% are composed by proteins, and almost 10% are related to lipids. So seeds are important and a good source of energy. We can use for humans, but especially for the plant development. And different of the primary metabolites, the secondary metabolites are natural compounds produced by plants and microorganisms when they are necessary. So they need some stimuli to have some expression and to be used. So they are mainly responsible for the plant environment interaction, adaptation, and protection against biotic and abiotic stresses. So here I show you some of the most important chemical classes that we can find in literature sources related to Trigonella fenugreek species. So we have the non-polar or the volatile compounds here represented by eugenol and linalool. So they have a very interesting biological activities, antioxidant, antimicrobial, antiseptic, anti-inflammatory. Also, we have phenolic compounds here represented by the flavonoids. The most reported compounds are the quercetin, rutin, also the isomers vitexin and isovitexin, and also a panel of interesting biological activities already report for them. Talking about the nitrogen-containing compounds, we have the alkaloids and modified amino acids. So the most remarkable amino acid reported is the 4-hydroxyisoleucine, which has been investigated for a long time to its anti-diabetic property. Also, alkaloid has the same property like a trigonelline, which has been also investigated for its anti-diabetic activity. The saponins are also always reported of one of the major compounds isolated and investigated from Fenugreek species, especially from the seeds, where is a promising source of the aglycone, the sapogenin diogenin, which has a wide panel of interesting biological activities. So, as you can see, Fenugreek species is a plant widely used for many purposes and contain a variety of different chemical classes of biological compounds, and which contribute to numerous health benefits. So here are some of the most common chemical classes which we can find: alkaloids, amino acids, coumarins, flavonoids, saponins. And if you look, the same compound can act in different infirmities or diseases, and more than one compound present in the species can act as a kind of synergism to against a problem. So Fenugreek can be considered as a phytocomplex. A lot of different compounds with different polarities and chemical classes that can act together in synergism and adding some different properties against the the diseases, against the, the hypoglycemic or some against some toxic effects, and they can thus how can I say decrease the dose necessary for the good effect, as well reducing the possibility of resistance. So we have here a multiple possibility of mechanism of action by different structures, different molecules, then can acting together in a phytocomplex against many different molecular targets. So it's a very interesting species. But why the plant produce the secondary metabolites? So here is an example, and the plant, they can run or escape from predators, from the UV light, from some fungi, bacteria, or attack from herbivore attacks, as well to attract the pollinator. So they need to produce some compounds, some natural compounds to attract the pollinators, to call their attention by different colors, to protect the young leaves from the impact of UV light. And all these reactions come from some gene stimulation or gene expression, and the stimulation of the activity of enzyme, specific enzymes that in the final of the reaction will lead to a different phenotype, that which the product is the production of different kind of metabolites. So the production of metabolite is the final chain of the gene stimulation and gene expression. So they are all complex mechanism together of stimulation for resistance, for adaptation, and process of genetic expression to final different phenotypes. So depending on the place, the environmental, and the stresses, biotic and abiotic, you can have different products. So for humans, the interesting thing is the pharmacological activities for us, but why the plant produce is for the plant defense. So they need the anti-UV properties and anti-insects as chemical deterrent, hormone, or pigment for protection. Here I show you some important secondary metabolite pathways. So here I show you the fourth most important for terpenoids, for phenolic compounds. So we have the mixed biosynthetic pathway, and also for just one, like for nitrogen containing compounds, like some aromatic alkaloids, they come from just one biosynthetic pathway, like shikimate pathway, while flavonoids need to the mix of two different biosynthetic pathway, shikimate and acetate malonate. And terpenoids can be produced by two different pathways. So the activation of inhibition of some of biosynthetic pathway will depend of the interaction and the stress which the plant is supporting in the environmental. So here shows some of the interesting compounds we can find in the Fenugreek species. We starting from nonpolar compounds. Here I present to you some of the most reported compounds isolated and characterized from essential oils. Here we have terpenes and some terpene derivatives. So we have the amiterpenes represented by the isoprene derivatives, the 2-methyl-2-butane-1-ol, and the other derivatives. We also have the monoterpene, linalool, which is a volatile compound, very popular and common reported as a monoterpene alcohol, essential oil, and it have a good fragrance to the essential oils. And here we have also the sesquiterpenes, compounds which are composed by 15 carbons, a little bit less volatile than the monoterpenes, but important to the Fenugreek essential oil composition. We also have the phenylpropanoid compounds. They come from shikimate pathway, and they are particularly characterized by the structure composed by phenyl, C6, and other 3 carbons. So the basic structure is composed by a C6, C3 in its framework. So here we have the 3 most known phenylpropanoids presenting essential oil of Fenugreek. So here the amiterpenes, monoterpenes, the sesquiterpenes, and the phenylpropanoids as volatile compounds present in Fenugreek essential oils. We also have some polar compounds which are very known and very important for antioxidant and also for anti-diabetes activity once they can scavenge the reactive oxygen species. So the flavonoids, as several different flavonoids have been isolated and characterized in Fenugreek's leaves and seeds. So here I show you on the top the flavonols, which are characterized by the oxidation C3 of the ring C, and on the bottom the flavones, the reduced C3 flavonoids. Both have shown interesting capacity as antioxidant, and it then has several important works related antioxidant, antitumoral, and anti-diabetes properties. So here I just present a resume about the mixed biosynthesis of flavonoids. So they are characterized by 3 different rings. So 2 rings are phenyl rings, the A and B, and the 3rd ring is a heterocycle ring. The ring B is here highlighted in green because it's come from shikimic acid pathway, so from the phenylalanine amino acid, and it brings the structure of phenylalanine amino acid, so C6C3, which is added 3 molecules of malonyl coenzyme A to generate the ring A. So the ring C, the heterocycle, is after closed by nucleophilic attack. So when the ring B is linked in position 2, is a common flavonoid. If it occurs some rearrangement reaction and it brings to the position of 3, carbon 3, it is considered as a isoflavonoids. So other important phenolic compounds characterized in Fenugreek species, mainly in the seed, but also can occur some of them in the leaves, are the pterocarpans. So they are considered some derivative from flavonoids. Here you can see the ring A, the ring B that was modified from the place from C2 to C3. Here in the center, the heterocycle ring, the ring C. A variety of different coumarins. Coumarins also have the structure of basic from shikimate pathway characterized by C6, C3. But here we have a lactone ring that was closed, and they presented a lot of different interest in biological activities. Also, from shikimate pathway, we have the by oxidative coupling. These structures are linked together, combining and leading to different kind of lignans, which are considered a very interesting biological compound. As I told you before, they have a lot of different phenylpropanoid compounds. These nonpolar phenylpropanoids can be found in essential oil, but also this structure can be linked to sugar, like here in the case of chlorogenic acid being part of polar fractions or extracts. Also, we have the stubenoids, which are have a close structure from the flavonoids. In the flavonoid case, we have 3 different rings. Here we have just 2 rings, not with a distance of 3 carbons, but here we have just 2 carbons separated these phenyl rings, but they are very important to Fenugreek because recently many works have highlighted the importance of stubenoids to insulin stimulation and diabetes potential control. So when you talk about the notable compounds from Fenugreek, we must to regard the phytosterol saponin. So here I present you the aglycone, the diogenin, sapogenin. So it's called, the aglycone is called sapogenin because it's sugar-free. It's a kind of spirostanol type because of the, of the last heterocycles. And here we have some of the most common isolated and characterized compounds. So imogenin, tygogenin, new tygogenin. So they are all aglycones, no sugar linked to the structure. So they are considered sapogenin. When these compounds are linked to different kind of sugars, they get different properties. So they have a long part which is non-polar with a very polar part has some amphipathic properties. So here they can have 3 different kind of sugars, 4 different kind of sugars, and linked in different ways. So the isolation of those compounds is challenging, and the NMR characterization, for example, is it's a very, very hard challenge. But this compound is very important for Fenugreek species because of this. Diogenin is a plant steroid that is very used since ancient times for centuries in Chinese traditional medicine, and it's related to menstrual cycles, menopause, and inflammation and infirmities. So they use the plant containing diogenin from a long time. In 1940s years, they have studied the wild yam, the species Discoria villosa, and as a promising source of diogenin. So diogenin, if you look here, why this molecule, this compound is so important, because its structure is quite similar to the progesterone hormone. So the researchers in the past realized that it was possible industrially to obtain the hormone by the degradation of diarginine. So this compound has economic value for pharmaceutical industry. So talking about the nitrogen-containing compounds, several alkaloids and amines were isolated from Fenugreek, but two deserve to be noticed. One of them is trigonelline, and one of the most important is the 4-hydroxyisoleucine. Both compounds present some interesting biological activity for insulin production and diabetes potential control. So here we're gonna talk just a little bit about the chemistry of 4-hydroxyisoleucine. As you can see, this molecule has 3 chiral centers, and this molecule was correlated to possess some insulinotropic biological activity. So it was verified that it can ability to stimulate the production of insulin. However, in nature is not presented as an enantiomeric total purity. It is found in a mixture of 2 diester isomers. So the major isomer is 2S, 3R, and 4S configuration. Found in 90%, while the minor compound is found in 10%, is 2R, 3R, 4S configuration. So how the industries sell this product? So due to the difficulty of isolating the totally pure compounds, they sell the products containing an enriched fraction of Fenugreek, and it still has potential anti-diabetic activity. So here is just an example. The product under the trade name Sugar Hill. So the major standardizer extract is Fenugreek extract, and other potential extracts here together in a complex phytocomplex. So as the last compounds we have here also the soluble fibers. These soluble fibers are considered very also important for the industry, because it is a polysaccharide with rigid hydrophilic backbone. Polymannose and galactose unite. So depending of the species, you can have different you can have a different ratio and distribution of the galactose and mannose in this structure. But in Fenugreek, the galactose mannose ratio is 1:1. So this compound is found in nature. It is usually found in cell wall released by, for example, fungi, the Aspergillus species, and it is has some industrial value since it's used in food as stabilizers, and it's also used in ice cream and salad dressing to improve texture. So I'd like to thank you all, especially Professor Hodaber Baham Sultani, for the invitation. If you have any doubt, you can send me an email. Here is my team in TechBio in Fiocruz Foundation. So I hope you have a very nice symposium. Um, um, متشكر. Thank you very much.
So we appreciate Dr. Marcus' lecture about the phytochemistry of fenugreek. Some compounds similar to those introduced by Dr. Bush was also explained here. So it shows which components of fenugreek are really industrially important. So as the third speaker of this panel, I would like to invite Dr. Nazlin Amazi, Assistant Professor of Nutritional Sciences, Diabetes Research Center, Endocrinology and Metabolism Clinical Sciences Institute, Tehran University of Medical Sciences, to talk about the impact of fenugreek in metabolic syndrome. Hello, everybody. I would like to share the findings of one of my systematic review and meta-analysis of the effects of fenugreek and the features of metabolic syndrome in adults. Metabolic syndrome is a cluster of metabolic disorders, including high serum triglycerides, high blood sugar, high blood pressure, abdomen, also increase the risk factors, diseases including diabetes and cardiovascular for metabolic syndrome, and currently one biochemical medicine for treatment of complementary therapy, and there are an excellent source. Sorry for the interruption. I couldn't change my slide. I think that there is a delay in changing my slide. Sorry for this. I think that it's much better to continue. Plants are an excellent source of chemicals for traditional medicine, and they often include bioactive mixes of varied active components with possible synergetic effects. A remarkable number of medicinal herbs are found to be helpful for treatment of diabetes, and some of them can be potentially targeting other risk factors linked with metabolic syndrome.
So due to limited studies, studies with the highest level of evidence, WHO recommends the researchers to do further studies on medicinal herbs to clarify the efficacy and adverse events of medicinal herbs. Okay. One of the medicinal herb is fenugreek. As all the speakers talked about the components of fenugreek, we found that antioxidants and anti-inflammatory components of fenugreek can play a key role in their positive effects, particularly for treatment of diabetes, dyslipidemia, CVD, obesity, and some types of cancers. When we decided to do this systematic review and meta-analysis, to the best of our knowledge, there was no systematic review and meta-analysis on this, and we aim to examine the effects of fenugreek and the features of metabolic syndrome as a primary outcome. And our secondary outcome was other metabolic disorders, including anthropometric indices and HDL and LDL cholesterol. So for the systematic review, we search for electronic databases plus reference lists of all eligible papers and relevant reviews. And we included clinical trials, so with a control group, which was conducted on adult population, and they reported at least one of the features of metabolic syndrome in mean and standard deviation or standard error. For this systematic review and meta-analysis, we didn't include the studies on a specific populations such as athletes, children, pregnant, and also studies which was a combination of fenugreek with the other herbs components, monomeric ingredient were not included. As well as this, gray literature were excluded. The protocol of this systematic review and meta-analysis has been registered in the PROSPRO as well. In the next step, after doing systematic review, systematic search by two independent researchers and screening the publication to reach eligible papers, they were extracted. All important data were extracted, as you can see in the yellow box on the right side of the slide. And then quality assessment was conducted. And at the end of the process, we did meta-analysis for reaching final decision. In this study, the JADOT checklist was used for quality assessment, and this checklist is given 5 score, from maximum 5 score to the articles. And when a study had at least 3 score, they were considered as high quality. Otherwise, they were classified as low quality study. Based on Cochrane textbooks, so we did data synthesis, and heterogeneity more than 50% was considered as high heterogeneity. Apart from main analysis, we did some complementary analysis such as sensitivity analysis, publication bias, fill and trim whenever was needed. In this slide, you can see the diagram of a screening process. As you see, eventually we reach 12 clinical trials. As one of our extracted data was related to adverse effects of fenugreek, we found that in just 4 studies, mild gastrointestinal disorders were reported, and only in 3 subjects, a specific urine smell was reported as well. No serious side effects was mentioned in all 12 clinical trials. And in one clinical trial, the effects of fenugreek on liver enzymes were examined. And after 8 weeks, no significant changes were found on these metabolic parameters. In this slide, you can see the effects of fenugreek on glycemic status. As you see, fenugreek can decrease the serum levels of FBS by around 30 milligram per deciliter. And however, the heterogeneity was high. And about hemoglobin A1C, although the reduction was statistically significant from clinical point of view, it was not remarkable. It's notable that publication bias for all variables were conducted, and we found no publication bias, and our sensitivity analysis showed that our findings are robust. In this slide, you can see the effects of fenugreek seed on lipid profile. As you see, serum levels of triglyceride and HDL didn't reduce after taking fenugreek seed. And regarding HDL, high heterogeneity was also found. So it's so important to do more clinical trials to confirm or reject the effects of fenugreek on these metabolic parameters. And the effects of fenugreek seed on total cholesterol and LDL cholesterol has been shown in this slide. The diagram and the feature of total cholesterol is after subgroup analysis based on the quality, because when we pulled all low and high quality studies, we found that fenugreek can decrease serum levels of total cholesterol, and after focusing only on high quality studies, we found that this reduction is not significant. However, the effects of fenugreek on LDL cholesterol was significant, and the reduction was around 11 milligram per deciliter. As one of the features of metabolic syndrome is waist circumference, unfortunately, we didn't found any clinical trial which reported this anthropometric index. And as a secondary outcome, we reported BMI, which is a kind of metabolic indices. As you see, fenugreek cannot reduce the BMI, but only 2 clinical trials were included, although the heterogeneity was the least, but it seems that more clinical trials are needed to confirm the efficacy of fenugreek on BMI. After the publication of 2023, another systematic review and meta-analysis, fenugreek can decrease FBS, triglyceride, HDL, waist circumference, and systolic blood pressure. In our study, no clinical trials were found which examined the effects of fenugreek on waist circumference, and due to high heterogeneity of 2 clinical trials which reported blood pressure, we couldn't pull this, and meta-analysis was not conducted for this variable. But as you see in this systematic review, systolic blood pressure was reduced after taking fenugreek, but from clinical point of view, it was not significant. In our systematic review, we report after doing subgroup analysis by dosage, we found that the dosage more than 10 gram per day can reduce hyperglycemia. And in this systematic review and meta-analysis, after doing dose response analysis, they reported that the dosage of 15 gram per day had the highest increasing effect for HDL and the highest reducing effect for FBS and triglyceride. Although precise mechanism for the effects of Fenugreek on metabolic disorders have not been fully understood, it seems that mostly it is related to antioxidant and anti-inflammatory properties of its ingredient. For example, evidence showed that Fenugreek can decrease inflammatory and oxidative parameters, insulin resistance, body weight, glucose and lipid absorption, and it also can increase insulin sensitivity, activate beta cells, and affect C-peptide, inhibit glucose transport, modulate GLP-1, and inhibit intestinal lipase action and reduction hepatic lipoprotein production. As a conclusion, we can say that fenugreek seed as an adjuvant therapy may reduce serum levels of FBS, LDL, and hemoglobin A1C. However, due to high heterogeneity in particularly glycemic status, findings must be interpreted with great caution. And further placebo-controlled clinical trials with larger sample size and high quality are needed to confirm the efficacy of Fenugreek as a complementary therapy for metabolic syndrome. At the end, I would like my dear colleagues from Iran, Australia, and the USA for their kind collaborations. Thank you so much for your attention.
Well, thank you very much, dear Dr. Namazi, for asked about the anti-diabetic dose of per day of the seeds can have anti-hyperglycemic properties. And actually, meta-analysis are those types of studies which provide the highest level of evidence. So they are well, sorry for the disruption. As the last speaker of this panel, I would like to introduce Dr. Elham Akhtari, Associate Professor of Traditional Persian Medicine from Iran University of Medical Sciences. She was unfortunately unable to deliver her lecture online, but she kindly recorded her lecture regarding the effects of Fenugreek in gynecology and obstetrics, which is based on one of the recent publication in Journal of Herbal Medicine. with the same topic. It was a collaboration between Dr. Akhtari, Dr. Marcus, and me. So as you all understood during this symposium, Fenugreek is an important medicinal plant for hormonal changes. And in this presentation, Dr. Akhtari explains about the role of this plant in gynecology and obstetrics.
Ladies and gentlemen, today I would like to present to you the evidence-based case for fenugreek as a treatment option for gynecologic disorders. Fenugreek, a plant-based herb, has been used for centuries to alleviate symptoms of various gynecological conditions. In recent years, numerous randomized controlled trials have been conducted to investigate the efficacy and safety of fenugreek in these conditions. This presentation will review the findings of these RCTs and discuss the implication for clinical practice. Our outline. The first, introduction, mechanisms of action and cautions, a little, and the RCTs and evidence-based medicine, fenugreek and gynecological problems, and at the end of our story, conclusion. A natural herb with a rich history. Yes, fenugreek. Fenugreek has been used in traditional medicine for centuries with a rich history of use in various cultures. Today, we'll explore the evidence-based case for fenugreek's benefits for gynecological disorders. In Persian medicine, fenugreek is called holbe or shambalile and is an important medicine in gynecology. According to Treasury of Medicaments, it can induce menstrual bleeding and therefore is used in amenorrhea. It is also a galactogogic in oral use. It is sometimes administered as vaginal suppository and sitz bath. According to canon of medicine, fenugreek can be used for uterine pain, inflammation, and intrauterine adhesions. In India, fenugreek is commonly known as holba or methi and has been widely used by Unani physicians for treating various gynecological problems. Its oral dose ranges from 3 to 7 grams and selected usual dose for enema is 70 milliliter. In Indian medicine, fenugreek is used alone or in combination with other medicinal plants in the form of decoction, enema, pessary, and sitz bath for the treatment of obstetrical and gynecological problems. It is also mentioned in Ayurveda to be a blood tonic which can be used in anemia due to menstruation. A little about mechanism and cautions. Although fenugreek is a popular plant material which has been widely used in culinary and medicine, it's not suggested to be used routinely due to some safety considerations. It is a rich source of phytoestrogens and based on some research is not recommended for subjects with a previous history of estrogen positive cancers. You can see it in the pathology sheets, ER/PR positive in breast cancer. Additionally, it is demonstrated that certain phytochemicals in foods and medicinal plants can affect the pharmacokinetics of conventional drugs by inducing or inhibitory effects on drug metabolizing enzymes and transporters such as hepatic cytochrome P450 and intestinal P-glycoprotein. Therefore, concomitant use of fenugreek when conventional drugs may affect their efficacy. There are some preclinical reports on the pharmacokinetic and pharmacodynamics interaction of fenugreek with clopidogrel and metoprolol. Another concern arises from the use of fenugreek in pregnant women due to the reported congenital malformation, anti-implantation, teratogenic, and abortifacient properties. So it should not be taken during pregnancy. This is also in line with the recommendations of different traditional medicines, such as Persian medicine, which bans the intake of emmenagogue plants like fenugreek during pregnancy. In Persian, like eating قرمه sabzi, it's a routine food. Additionally, as fenugreek is a member of the legume family, it should not be taken by those with a past history of allergy to other plants of this family, including peanut, due to the possibility of cross reactivity. A short look on traditional use of fenugreek. From the past till now, scientists uh, uh, noticed scientists noticed to Fengreek and its applications uh, to manage gynecological problems, from galactogogue, emmenagogue, till uterine pain, inflammation, adhesions, as a blood tonic in anemia, amenorrhea, cold distemperament of uterus, meteoritis, endometrial sclerosis, vaginitis, vaginal white discharge, amenorrhea, and the other applications. We start with breastfeeding in RCTs. 2015, Qasemi and uh, uh, their colleagues, as a title, the effect of herbal tea containing fenugreek seed on the signs of breast milk sufficiency in Iranian girl infants, showed us galactogogic properties of fenugreek. They used 7.5 gram of fenugreek infusion 3 times a day in 78 nursing mothers. 4 weeks of intervention showed significantly higher urination, defecation, and breastfeeding in infants in the intervention group compared with the control group. Another randomized control trial used macerated extract of 7.5 fenugreek. It was by Ravi 2020. They used once daily fenugreek, which is 1/3 of the administered dose in the former study. After 1 week of administration, a significant increase in milk production as well as urination and weight gain were observed in infants upon supplementation with fenugreek, which shows higher milk intake. Menopause. Menopause is defined as the everlasting cessation of menstruation, usually after 12 months of amenorrhea at the end of reproductive years of women. Menopause has side effects, you know them, like sleep disturbances, hot flashes, osteoporosis, vaginal dryness, and joint pains. A clinical trial by Steels 2017 showed us with daily 600 milligram of fenugreek powder in 115 postmenopausal women also reported significantly decreased menopause symptoms. The evaluation was based on the menopause specific quality of life questionnaire, which showed a remarkable improvement in psychosocial, physical, vasomotor, and sexual symptoms. Mazalzadeh, uh, um, 2020, uh, had another example. RCTs using the same fenugreek preparation was performed in 60 postmenopausal women with vaginal atrophy, in which a significant elevation in vaginal maturation index was observed in comparison to baseline values. Wu uh, um, has another RCTs 2020. It seems that the oral intake of fenugreek extract with a daily dose range of 500 to 1000 milligram is effective for the management of menopause complications. However, meta-analyses are needed to further confirm the efficacy of fenugreek in this condition. The other one is dysmenorrhea. Painful menstruation is defined as a severe, painful, cramping sensation in the lower abdomen that is often accompanied by other symptoms such as sweating, headaches, nausea, vomiting, diarrhea, and the other symptoms, all occurring just before or during the menses. Fenugreek is one of the most popular medicinal plants used to control these pains, which is also elevated in clinical studies. One of these RCTs uh, was 2016 performed a clinical trial in 60 women with primary dysmenorrhea using 1 gram fenugreek powder 3 times a day for the first 3 days of the menstruation period. The title was Clinical Efficacy of Fenugreek and Dry Coughing Therapy on Intensity of Pain in Patients with Primary Dysmenorrhea. The other one uh, mm, did by UNESC 2014. The title is Effects of Fenugreek Seed on the Severity and Systemic Symptoms of Dysmenorrhea. Uh, mm, this RCT support the beneficial effects of fenugreek supplements in controlling symptoms of primary dysmenorrhea with a dose range of 1,800 to 3,000 milligram per day without any concerning side effects, without any reported side effects. The other one is PCOS. So, Rope 2050 with title Efficacy of a Novel Fenugreek Seed Extract in Polycystic Ovary Syndrome did a RCT, single arm open label clinical study with 50 patients, received 1 gram per day of the supplement over 3 months, which resulted in a significant elevation of FSH and LH. There was a significant reduction in cyst size in for 36% of patients, whereas complete cyst dissolution was observed in 36% of cases. Sanhua with another study. They're using the same formulation. 107 volunteers were treated with 1 gram per day of fenugreek preparation. Upon 12 weeks of treatment, more than 40% of reduction in the size of ovarian cysts were reported, along with a significant improvement of LH to FSH ratio, as well as a decrease in prolactin and testosterone. The title was Beneficial Effects of a Novel Fenugreek Seed Extract in Women with Polycystic Ovary Syndrome. Considering the included clinical studies on PCOS, 1 gram per day of fenugreek seems to have a significant effect on the improvement of symptoms in patients with PCOS. Although these studies may seem insufficient to support clinical administration of fenugreek in PCOS, the provided data introduces fenugreek as a promising candidate to be further studied in randomized controlled trials. Conclusion. Fenugreek is one of the most important medicinal plants as a calm remedy for women's problems. It is a well-known and globally available plant that has been extensively studied in gynecological problems. So, in traditional and complementary medicine, it is frequently recommended in these cases, especially in PCOD and dysmenorrhea. Clinical studies support the use of fenugreek in several gynecological problems, including menopause, dysmenorrhea, and breastfeeding. So, this plant can be recommended as a useful supplement in such diseases. Safety issues should also be considered for the administration of it by physicians in order to prevent possible side effects. Future clinical and mechanistic studies are necessary to evaluate the safety and efficacy of fenugreek in other gynecological disorders. Thank you for your attention.
Well, we also appreciate Dr. Akhtari's lecture on the application of fenugreek in gynecological and obstetric problems. So we are 5 minutes behind the program. We're going to have a very short break, around 2 minutes, and then the third panel with oral presentations of the participants who submitted abstract will be continued. I would like to thank you all for being with us today, and please stay in the room to listen to the oral presentations in 2 minutes. Thank you very much.
Okay, I'm going to take it from here. Welcome back, everyone. I hope you have enjoyed the symposium so far. We're going to start the third section with Dr. Indira Dutta to kindly present her presentation. Dr. Dutta, you have the access and you can start your presentation. Dr. Dutta, are you hearing my voice? Could you please start your presentation? Okay. We're having your video and we're waiting for you to start. But we're not having your voice yet. So we're not having your voice yet. So I think it's better to, okay, just take your time. Okay, I think for sticking to the time, we're going to move on to the next speaker, but we'll get back to Dr. Indira after the first speech. So I'm going to invite Dr. Arshia Sultana. Dr. Arshia Sultana, if you're here.
Yes, ma'am.
Okay, very good. Hello. So
Hello, good afternoon. I'm audible to you?
Yes, totally audible.
Good afternoon.
Thank you. Go on, please, Mary.
Can I share my screen?
Yeah, you have the access.
Okay, good afternoon, all of you. At the outset, I'm thankful to Tehran University of Medical Sciences for giving this opportunity to present my oral presentation. So here, today my topic is therapeutic efficacy of triglona and hijama pilashut in primary dysmenorrhea. So a background. As we know, primary dysmenorrhea is the most common gynecological disorder in menstruating women. It is an important women's health issue with high prevalence, about 45% to 95%. It affects the quality of life in 60 to 90% of females, and we see that primary dysmenorrhea is more common in young children, affecting their successful education, psychosocial and cognitive development during the critical growth period of adolescent, and therefore significantly lowers the quality of life. So current evidence suggests that primary dysmenorrhea is associated with ovulatory cycles and is largely due to myometrial contractions induced by excess endometrial prostaglandins that are produced mostly in first 42 hours of menses. This local action of prostaglandins on the uterus is threefold. That is, it increases the basal intrauterine pressure, causes constriction of uterine arteries, subsequent tissue ischemia leading to pain, and finally increases the sensitivity of peripheral pain nerve endings. In Unani system of medicine, pain is a perception of contiguity afflicting the human body, which is an abnormal condition caused because of sudden and abnormal change in temperament, that is distemperament, and breach of continuity, that is tafarruq ittisal. These are the two main causative factors leading to pain. Sometimes the expulsive faculty of uterus becomes weak by obstruction formed by ghalis, that may be balgam or sada, that is balgam or black bile, which may be one of the causative factors of dysmenorrhea. Considering the present unconvincing scenario regarding the use of drugs and adverse effects thereof, researchers are pursuing the golden formula of turning to nature anti-ritualopathies. Fenugreek is one of the oldest known medical herb in the recorded history. Just now in previous lecture, we have heard that it is useful in dysmenorrhea, uterine inflammation, amenorrhea, licoria, etc. According to Unani scholars, dry cupping therapy, Peloda umbilicus, relieves dysmenorrhea, especially in young girls. In Unani classical books, it is said that fenugreeks have anti-inflammatory, antispasmodic, analgesic, diuretic, and immunogogue properties. So with this hypothesis, the objective of the study was to determine the efficacy of fenugreek and hijama bilashat in primary dysmenorrhea. The methodology of this study was, it was a comparative, prospective, single-center, 3-arm, open-labeled, randomized standard control trial conducted in National Institute of Unani Medicine between duration February 2010 and April 2011. Ethical clearance was taken from the institute and informed written consent was taken from the patient before enrolling in the study. The inclusion criteria of the studies were participants with primary dysmenorrhea aged between 12 to 30 years, having regular menstrual cycle. Both participants, married, unmarried, were included in the study. Participants with congenital anomaly of uterus, secondary dysmenorrhea, organic pelvic pathology, membranous dysmenorrhea, systemic illnesses, oral contraceptives, and other hormonal agents were excluded from the study. The procedure was patients with were assessed through complete history, physical examination. Socio-demographic data was also collected. All the participants who were included in the study, general and systemic examination was carried out, and if the woman was married, pelvic examination was also done to exclude pelvic pathologies. So, the percentage of pain reduction was calculated by this formula: baseline bar score minus mean pain reduction score of 3 consecutive cycles into 100 divided by baseline bar score. The outcome which we were taking was satisfactory pain relief was considered to be when percentage pain reduction was more than or equal to 50% and taken as non-satisfactory when the percentage pain reduction was less than 50%. The investigations we carried out in this trial was hematological urinalysis and specific investigations, pelvic ultrasonography. Here we can see that in pre and post test, we have done safety profile, including ALT, AST, alkaline phosphatase, blood urea, and serum creatinine. Intervention was divided into 3 groups: Group A, Group B, and Group C. Group A included 24 subjects. They were given text drug, that is 3 gram powder of fenugreek seed, that is 1 gram, 3 capsules containing 1 gram each twice daily from day 1 to day 3 of menstrual cycle. Group B also contained 24 number participants received same dose of fenugreek seed as Group A along with dry cupping. 3 cups were applied to 4.7 centimeters internal diameter and 2.1 centimeter cup internal diameter, which was applied below the umbilicus for 15 minutes on day 1 and day 2 of the menstruation. Group C included standard group receiving mefenamic acid 500 milligrams twice daily for 3 days. This was the location, 1 more cup for this, where the cups were applied. So duration of treatment was 3 months. Follow-up was after menstruation for 3 months and after treatment once after 1 month. Withdrawal criteria if they failed to follow the protocol or cases where adverse reactions were noted. Outcome was primary was for lower abdominal pain and low backache. Secondary outcome was systematic symptoms and safety evaluation. This was the regional analog scale for pain intensity used in this study. It's a color scale. I think so all of us know how to measure on this. So here sample size was calculated. We have taken 72 participants, including 20% iteration rate. This sample size was calculated from the previous study. Randomization allocation was 1:1:1, and open list of random numbers were obtained from computer-generated randomization list. The random allocation sequence was prepared in single blocks by means of single sequence of random assignment. The sequence was concealed from the receiver until the interventions were assigned using an open list of random number. Statistical analysis, we have used GPAT in this to do the analysis. This analysis included per protocol principle. Type 1 error was 0.05 and type 2 error was power of study was 80% with 95% confidence interval. The test used was ANOVA, as we have taken 3 arms in this study. The results, the mean age in the 3 group was 17.70, 19.10 and 18.05 years in the test, and group A, group B and group C. This is the consort statement. Flow of participant, we can see here we have seen for eligibility 120 participants, in which 33 participants refused to include in the study, and 15 participants were excluded. And we can see that only 20 participants who have taken the whole medication has been included in the trial in all 3 groups. So we can see the results, comparison of VAS of lower abdominal and backache. Here we can see that all the participants had severe abdominal pain during menses and was comparable before test. And we can see that at the third cycle, the VAS score has decreased to 0.7, 0.86 and 0.74. We can see that they were not significant in 3 groups, showing that the 3 groups were equally effective in the treatment. Then not only that, we have seen associated symptom fatigue. We can see that most of the participants had moderate fatigue in group A, whereas in group B it was mild, and in group C we can see it was 6. So we can see that most of the participants had fatigue during menstrual pain. Then in anxiety, headache also, we see that 5 participants had headache. 6 participants had headache in group A, but after treatment only 2 participants had mild headache and 18 were not having any headache. Here again we can see 8 participants had anxiety, and here we can see nausea, vomiting was present in 4 participants. So we can see that associated symptoms were not that much during menstruation pain. Then comparison of biochemical parameter among 3 groups, we can see that it was comparable before and after test in all 3 groups, and it was not significant before and after as well as intergroup or an intergroup comparison. Then safety profile, again we can see that it was comparable, and we can see that it was not statistically significant in all 3 groups, intergroup as well as intergroup comparison. So results indicates that percentage reduction in lower abdominal pain was 66.89% in group A, 66.49% in group B, and 62.88% in group C by the end of the treatment period, showing that all the 3 groups were equally effective. Safety assessment showed no adverse events were reported in both groups. So as already we saw that Fenugreek have analgesic, anti-inflammatory, antipyretic, antioxidant and antispasmodic properties. So mainly because we know that menstrual pain is caused because of prostaglandins, and usually we give NSAIDs in modern medicine. Likewise, we can say that Fengreek is also having anti-inflammatory and analgesic activity, which is seen because of presence of flavonoids, because in previous lectures just now I saw how this medication is acting. So the presence of flavonoid and saponins, bioactive components, we are able to see anti-inflammatory and analgesic effect. Moreover, the linoleic acid is converted in dihomo-gamma-linoleic acid, which competes with arachidonate for oxidative enzyme, thereby reducing production of cyclooxygenase products derived from it. That means it's showing that how it acts on cyclooxygenase and reduces the inflammation. The benefit of anti-inflammatory effect of Fenugreek seed is directed towards modulating one's responsive to PG levels. Apart from this, Fenugreek also contains magnesium, zinc, thiamine, and vitamin E, which are also helpful in dysmenorrhea. Then, cupping, as we know, cupping, the main action of dry cupping is imalemava, that is shunting of blood flow away from the viscera. So we see that when we apply cup on the lower abdomen, we are able to see that it decreases the congestion in the pelvic floor according to the hypothesis of Unani system, and suppresses the prostaglandins, thereby causing reduction in pain and promotes release of beta endorphins, providing endogenous analgesia. Second hypothesis can be it stimulates A-beta sensory fibers from the peripheral tactile receptors, which depresses the transmission of pain signals, which results from local electronegating or activating activating the immune system, enkephalin secretion, neurotransmitter release, vasoconstriction and dilatation, and the gate theory, stating that how the pain is reduced by applying the cups. Still, till date, none of the clinical intervention studies evaluated Fenugreek and cupping therapy in dysmenorrhea. It was a randomized standard control study with good compliance. Allocation was done by randomization and interventional trial, including early to late adolescent age and Paris women with moderate to severe dysmenorrhea. Limitation was it's an open label and small sample size. Further recommendation, we recommend phase 3 trials. For longer duration, the efficacy of Fenugreek seed and dry cupping therapy can be appraised in secondary dysmenorrhea. Moreover, to find the specific role of fenugreek seed and dry cupping therapy on prostaglandins, the levels are to be estimated in menstrual blood during menstruation before and after trial. Fenugreek is proven to be gastroprotective, therefore it can be recommended not only in dysmenorrhea but also in other types of painful condition instead of NSAIDs that causes gastric mucosal damage. So conclusion, fenugreek seed alone and fenugreek seed with dry cupping therapy is a suitable option in reducing severity of pain in primary dysmenorrhea. Fenugreek seed and dry cupping therapy is cost-effective, safe, and well tolerated by women. Thank you.
Thank you very much, Dr. Aisha. It was very, very interesting. And I would like to say that this study was very valuable, as dysmenorrhea can really disturb women's routine life worldwide. So it's a very good idea to try to control the pains with complementary medicine, with herbal medicine, which are rather safer. And so I think it's very, very valuable. Thank you very much.
Thank you, ma'am.
Presentation. So let's go to the next speaker. I would like to invite Dr. Indira Dutta. I don't know if your problem has fixed or not, but if you're ready, we're here to take your presentation. I'm not sure if you're in the room. Okay. So I hope the technical issues has been fixed. Dr. Indira Dutta. Very good. Thank you. I think you have shared your screen and it's visible. Okay, we're not hearing your voice yet. Okay, let's wait for a minute. I hope the technical issues will be fixed. And I want to kindly ask other presenters to be ready for their speech. Okay, now your sound is audible. I think you can start your presentation.
Am I audible right now?
Hello, yes. You have your voice.
Okay. Good afternoon, everyone. Myself, Indira Datta, and I would be presenting on the topic Fenugreek seeds in skin health and cosmetics, a review of topical and oral applications.
Okay, sorry. Are you sharing your slides? because it's not visible.
It's not visible?
No,
Okay.
we're not having your slides. Okay, thank you.
Okay, thank you. So today I would be presenting on the topic Fenugreek seeds in skin health and cosmetics, a review of topical and oral applications along with my co-presenter Preetha Dev. Introduction. Fenugreek, also known as Trigonella phoenum gracium, an ancient medicinal plant which has been utilized for its medicinal properties for centuries, as reported by a study by Ruvali et al. 2022. Fenugreek seeds have been shown to hold appetite suppressing properties and can help control hunger and reduce food intake. It is rich in antioxidants.
Sorry to interrupt you. I think it's better to change it to full screen. And also, we're still on your first slide. I think you've moved to the second one. So is it possible for you to make it full screen? Make it bigger? Okay, now we're having the slide of introduction.
Okay. Can I continue right now?
Yes, yeah, go on.
Thank you. So as I was saying that fenugreek seeds are rich in antioxidants including polyphenols and flavonoids which combat oxidative stress and reduce the damage caused by free radicals. Therefore protect cells from aging and lower the risk of chronic diseases. Traditionally, fresh fenugreek leaves paste applied over the scalp regularly before bath, and it also helps in the hair growth, preserves natural color, keeps hair silky, and also cures dandruff. Methodology. In this entire presentation, I have used um, studies selected for review 8 and record excluded are 95. Record included through database searching was 100. Role of fenugreek seeds in skin and cosmetics. In skin health, fenugreek seeds are known for their anti-inflammatory, antibacterial, and moisturizing properties. They are particularly effective in managing conditions like acne, eczema, and skin irritation, as well as for their potential anti-aging benefits. Fenugreek seeds contain compounds like mucilage and diosgenin, which help hydrate the skin and reduce wrinkles and fine lines. In the cosmetic industry, fenugreek seed extracts are often used in creams, serums, and face masks to improve skin texture, boost hydration, and promote a glowing complexion. The seeds can also be ground into a fine powder for exfoliation or mixed with natural oils to create nourishing skin treatments. With their combination of soothing and rejuvenating properties, fenugreek seeds are becoming an essential ingredient in both traditional and modern skincare routines, offering a natural and holistic approach to achieve a healthier skin. According to Kim et al. 2018, this research focused on the photoprotective effects of fenugreek seed extract against UV-induced skin damage. The authors found that the extract induced the expression of matrix metalloproteinases and increased the collagen synthesis in UV-exposed skin cells. Layers of skin. Keratinocytes. Fenugreek seed extract may help protect the keratinocytes, the most common cell type in the epidermis, from oxidative stress, and it also improves the skin's barrier function and overall health. Fenugreek has also been shown to stimulate the fibroblast activity. This can lead to increased production of collagen and elastin, which are crucial for maintaining skin elasticity and reducing the appearance of the wrinkles. Melanocytes. Some studies suggest that compounds in the fenugreek seeds might help to regulate melanin production in melanocytes. Langerhans cells, while not well extensively studied, there's some indication, according to some studies, that fenugreek's anti-inflammatory properties might influence Langerhans cells, potentially modulating the skin immune responses. Fenugreek's potential effects on the sebocytes, that is, the sebum producing cells, are not well documented, but its anti inflammatory properties might help in managing conditions like acne. Antibacterial properties. In this study, boiled and methanolic seed extracts in different concentrations were tested against acne and pimple causing bacteria, namely P. acnes, S. aureus, and S. epidermidis, using agar-well diffusion and disc diffusion techniques. The study proved that methanolic extracts having a better antimicrobial response against these bacteria. Antifungal properties. Aqueous and ethanolic extracts of the fenugreek leaves exhibited activity against a clinical isolate and commercial strain of Malacia furfur as reported by a study by Kulkarni et al. 2020. The extracts were also found to be active against other pathogenic fungi such as Aspergillus niger and Candida albicans. Qualitative and quantitative phytochemical evaluation of the aqueous extract showed a predominant presence of flavonoids. The gel formulation of 30% aqueous fenugreek leaf extract was developed and optimized by using sodium alginate as a gelling agent, and this formulation showed a good physiochemical characteristic and retained activity against M. furfur and dandruff. Now I would like to hand over the further slide to my co-presenter Pritha Dev.
Hello everyone, this is Preetha Deb and I'm going to continue the rest of the presentation. Anti-wrinkle effect. Fenugreek seed possesses antioxidant effects and contain a mucilage which has soothing properties. It promotes skin healing, whitening, moisturizing, skin soothing, and anti-wrinkle effects. It can be concluded that stable watery oil formulation using extract of fenugreek seed and paraffin oil can be reformulated and used to reduce skin melanin contents and to increase skin moisture content without causing any skin irritation. According to Jamal et al., this systematic review examined the potential of fenugreek seed extract as an anti-aging agent for skin. The authors concluded that fenugreek seed extract shows improvement in skin elasticity and reducing wrinkles due to the antioxidant and anti-inflammatory properties. Anti-cancer properties. Fenugreek seed contains saponins, flavonoids, and alkaloids that exhibit anti-inflammatory and antioxidant activity by reducing cytokinins, which are pro-inflammatory compounds that can lead to inflammation and disease. Diosgenin, a steroidal saponin found in fenugreek seeds with structure similar to estrogen, has been shown to have proapoptotic and anti-cancer activities. Topical application. Based on study, herbal hair oil was prepared including hibiscus flower, leaves, and fenugreek seeds in varying concentration by preparation techniques like direct boiling, paste, and cloth method, and were tested for hair growth activity. Methi and hibiscus oil, respectively, with different concentrations, were applied in the same pattern on the back of the albino rats for 15 days. Methi oil, that is fenugreek seed oil, showed growth in 9 to 10 days at a concentration of 7 to 8%. According to Rashid et al., the study examined the effects of topically applied fenugreek seed mucilage on skin hydration and barrier function. Findings indicated that the mucilage significantly improved skin moisture retention and enhanced the skin barrier properties. Aids in collagen production and inhibits skin aging. Based on the study by Usheria et al., the inhibitory concentration of fenugreek seed extract was lower than that of ascorbic acid, indicating its potential as an anti-aging agent. As per study reported by Singh et al. And Tamara et al., it investigated the effects of fenugreek seed extract on human dermal fibroblast. Results suggested that the extract enhanced collagen production and protected cells against oxidative stress as it contains an active compound called ruetin, indicating potential anti-aging benefits for skin. It was also reported that sun exposure would reduce ascorbic acid content by 84.94%. There was better retention of nutrients in the fresh leaves of fenugreek. Anti-inflammatory and wound healing properties. According to Muhammad et al. And Sharma et al., these studies investigated the anti-inflammatory and wound healing properties of fenugreek seeds extract on human skin cells. The extract was found to reduce inflammation markers and promote faster wound closure in vitro skin models. Fenugreek seeds contain fatty acids which build collagen and promotes wound healing and maintain elasticity. Lastly, I like to conclude the whole presentation by saying that Fenugreek seeds are known for its medicinal properties in type 1 and type 2 diabetes and hypercholesterolemia. Because of its antioxidant properties, fenugreek seeds have been shown to have positive impact on the liver and pancreas. Apart from this, fenugreek seeds also offer significant benefits for both hair and skin. They are rich in anti-inflammatory, which help in wound healing, anti-cancer properties, and also aids in collagen production and inhibits skin aging. It has antifungal properties, which helps to reduce dandruff and hair fall, thus nourishing the skull and promoting hair growth. This is a reference slide. Thank you.
Thank you very much. It was a very nice presentation, and the topical and cosmetics forms, they're all very, very interesting. I think it was somehow in line with the therapeutic effects of fenugreek, which was explained on the presentation about Persian medicine. I think Persian medicine also has mentioned that fenugreek is effective in acne, dandruff. It can heal wounds, and also it has anti-inflammatory effect. Thank you very much. Just a quick comment. I think it was better to explain the method part a bit more conclusively, but the rest was very good, and thanks for your presentation. So just let's move on to the next speaker. I want to invite Dr. Ashiru Aliyu, if I'm pronouncing okay. Is my pronunciation is okay? So Dr. Ashiru, if you're here. So, okay. So Dr. Ashiru, if you're with us, we're here to, we're ready to take your presentation. Dr. Ashiru, you have the access. And if you're ready, you can start your presentation. Dr. Ashiru Zainul Abedin. I think she's not having my voice. So we're gonna move to the next speaker, which will be Dr. Shahid Shihana. I'm gonna kindly invite you to give your presentation. Hello.
Good afternoon, everyone. Can you hear me?
Yes, yes, your voice is sound. Thank you very much. And we also have your slides.
Okay. Okay. Good afternoon, everyone. I'm Dr. Shihana, MD Scholar in Knowledge at Postgraduate Institute of Indigenous Medicine, University of Colombo, Sri Lanka. My topic is effectiveness of trigonal breast paste in the management of neuropathies. This is a case series from our clinic. According to Unani medicines, Wajah al-Asab, or the qadar means defined for the neuropathic pain, and qadar is termed for the sensory impairment like numbness and paresthesia. Ibn Sina also divided the neuropathy into several groups: qadar, paresis, paralysis, tremor, lakwa, and tasanoch. The pathophysiology, according to Rasi, qadar is the intila of ghaliz ahlat. That means the cold type of ahlat, and also the frequent exposure to cold which causes accumulation of blood in nerves and weakening them and affecting nerve conditions. According to modern medicine, approximately 2.4% of the population affected by the neuropathies, especially by the diabetic neuropathies. And most frequently encountered symptoms includes numbness, paresthesia, pain, weakness, and in severe cases, loss of deep tendon reflexes. And the today topic is the vulva, Fenugreek, trigonal for enum brachium. According to Unani medicine, it possesses several important actions like Munsich, Muhallilevaram, Mukaviyaza. And the objective of this study is evaluates the therapeutic potential of Fenugreek in alleviating various types of the neuropathies. The patients are from our hospital outpatient department. We conducted these studies among 14 patients between the ages between 35 to 70 years. And the individuals diagnosed with diabetic neuropathy, five cases, and carpal tunnel syndrome, 6 cases, and sciatic neuropathy, 3 cases. The main symptoms includes the burning and tingling in the paresthesia, dyskinesia, and also the sensory changes. The patient were diagnosed using nerve conduction studies, and these are the history explaining, and some of the patients are diagnosed using the electromyography, and the carpal tunnel syndrome diagnosed with the tire signs and financial signs, and the sciatic nerve compressions was diagnosed with using the MRI. The intervention, this is a very simple paste. We can also make it our home. The external application of paste made out of Fenugreek, Asafoetida, and aloe extract for 12 hours daily over 4 weeks. The method of preparation include first sibur was dissolved in hot water, and the badian and pulpa powder were added little by little until the consistency of the paste. And the outcome were assessed using the visual analog scale, neuropathic pain scale, and also the neuropathy disability scores. The VASI scale, the patients were asked to mark on the line at the point that they feel represent their current state of pain. The neuropathic pain scales includes these are the symptoms we assessed. And the neuropathy disability scores include the vibration perception, temperature sensation, prick sensation, and the reflexes. So the results includes among the patients, for the diabetic patients, before the treatment, we analyzed the VAS scale between 6 to 8, and after the treatment, it reduced between the 4 to 6. And for between the carpal tunnel syndrome patients, more than 9 before the treatment. And after the treatment, it was between 2 to 4. And for the sciatic neuropathy, the numbness patients, the VAS scale was more than 7, and after the treatment, it reduced to 4. And the neuropathic pain scales, that is more than 8 among the diabetic patients, and it was used more than 4 after the treatment. And carpal tunnel syndrome patients suffered more than 6 among with the neuropathic pain scale and reduced to 3. And for the sciatic patient, it was 8 and reduced to 4. And the neuropathy disability score also reduced with a higher percentages. So before the treatment, it was about 8 for all 3 groups, but it was reduced less than 6 after the treatment. But for the carpal tunnel syndrome, it is all after the treatment, it was 4, less than 4. And some studies also suggesting the neuroprotective effect of the Fenugreek, that they suggested the anti-inflammatory and antioxidant properties, especially the role of trigonelline. That is the major alkaloid present in Fenugreek seeds. And in their study, they administrate daily oral of trigonal extract, seed extract. So they found out the protection against thermal hyperalgia and deranged motor function test. Importantly, sciatic nerve cross-injury control rates showed significant reduction in motor nerve conduction velocity when compared with the normal group. And also they stated that modulation of sensory pathway and inhibition of cytokines release as one of the probable mechanism by the fenugreek extract against the thermal hyperexia in peripheral neuropathies. And also the anti-inflammatory effects of fenugreek extract were attributed and due to the trigonally, that is protection against neuronal inflammation. Another study by Abdul Bari stated the significant decreases in anti-inflammatory markers such as TNF alpha and IL-6. There's also another cause for the neuropathy. And according to Ali, neuropathy was induced in male mice pyridoxine intoxications, and they found that significantly increased the mean nerve conductions velocity comparable to the other groups. And another study also reported among the diabetic rates showed significant improvement in nerve function and reduction in neuropathic pain. And the neuroprotective effects were linked to the presence of diosgenin, an active compound in the fenugreek, which promotes nerve growth and reduces neuroinflammation. Another study reported the other ingredients. We also assessed the other ingredients other than the fenugreek, the SF-whitida, oleogum resins of the SF-whitida also responsible for the improved nerve conduction velocity and axonal regeneration in pyridoxine-induced neuropathies. And also the potassium channels in the vasoreduction effects of SF-whitida also help in reducing the neuropathies. And another research suggested that the polysaccharides in aloe vera may enhance antioxidant defenses and reduce hypoglycemia-induced nerve damages. And when comparing with among these three groups, fenugreek showed the more effect when compared for the neuropathies, reducing the neuropathies when compared with the other two ingredients. And in conclusion, this is one of the topical applications in our hospital. That is the fenugreek-based formulation. It can be an effective treatment for peripheral neuropathies. And the larger randomized control trials are needed to confirm these findings. And these are some of the references. And thank you very much.
Thank you very much for your presentation. I think it was a case series, but you focused on the underlying mechanism and made it and actually added value to this study. Thank you very much. It was very, very good.
Okay.
So, thank you. So I'm going to invite the next speaker, which again will be Dr. Ashiru Zainul Abidin, if you're having my voice. My colleagues has given you the access for sharing your slides and just unmuting yourself. If you're having my voice, you can start your presentation. Okay, Dr. Zainul Abidin, is it possible for you to start your presentation? So if you're not having my voice, if it's not possible for you to give your presentation, we're gonna wait for the very last presenter, which will join us very soon. We're gonna wait for 1 more minute and then we'll decide what to do next. So we're gonna wait for a couple of seconds and then we have two more speakers. I don't know if they're able to join us. We're gonna wait for a couple of minutes and see if they're able to give their presentation. So I think as our two last speakers are not able to give their speech, I'm gonna kindly invite Dr. Baram Soltani for conclusion remarks and closing ceremony. Okay, Dr. Ashiru, is it possible for you to give your speech? I think you have the access for unmuting yourself. Okay, hello.
Can you hear me?
Could you please speak a bit louder?
Can you hear me?
I have your voice, but it's not clear. So is it okay for you? Can you start your presentation? Okay, now we have your video. Yeah. So could you please share your slides? If you're ready, you can start your presentation.
Sorry, can you hear me?
Yes.
Okay. Sorry, let me start sharing the screen.
Could you please share your screen with us?
Yes, I will start sharing the screen now. Sorry. Can you see the screen now? Sorry.
Not yet. We're not having your screen.
Okay.
There's a green button with it's written share screen. I think you can just choose your presentation and that will be okay.
Sorry, I'm having technical issue.
Okay, I think you have started sharing your screen, but still we don't have your slides.
Sorry, I'm having technical issue with the network.
Okay. So I think it's better to move on to the next speaker. But after this current speech, if your technical problem has been solved, then we can have your speech. But if not, we need to move on because our time's so tight. So Dr. Mahnaz Karimi, I would like to invite you to give your speech. You have the access, so we can, okay, hello. You can share your screen. We don't have your voice, but I think your mic is open.
Hello, Dr. Ismailzadeh.
Hello, okay. So I think you can share your screen and start your presentation.
Okay.
Okay, we're having your screen. So we can start the presentation.
Problem. Susan.
I think there's a Fenugreek, yeah, file there. Okay. It's okay, I think you can start.
Hello, everyone. Good afternoon. I'm Mahnaz Karimi from Tehran University of Medical Sciences. I appreciate Dr. Bahram Sultan and Dr. Ismailzadeh. It's an honor to be here in this symposium. The topic of my presentation is a review of the effect of Fengreek, Tribunella Fenugreek on osteoporosis. As we know, osteoporosis is an epidemic disease by low bone density that increase bone fragility and bone fracture. This bone fracture increase increase mortality, morbidity, and decrease the quality of life and impose a lot of cost on society or individuals. This is an overview on bone metabolism. When the balance between bone resorption and bone formation is disturbed, osteoporosis occurs. So we know some factors such as menopause, aging, low bone mass index, or physical inactivity can aggravate this osteoporosis or osteopenia, and there are some available treatments such as hormonotherapy, bisphosphonate, raloxifene, denosumab, that they are so effective, but they are not recommended for long-term use because of some side effects, such as hormone therapy can increase the risk of cardiovascular diseases or ovarian and breast cancer, and some side effects such as osteonecrosis of mandible is seen by long-term use of bisphosphonate or denosumab. So it's important to pay attention to herbal medicine and complementary to discover new drugs. For example, there are a lot of plants that investigated the effect of them on osteoporosis. One of them is Fengreek, the scientific of Tribunella from Fabaceae family. There are some common names, for example, Holbe or Holba in Arabic, Chamberlain in Persian, Meti in Indian, and Fenugreek in English. We know the use part are leaves and seeds, and although it's native to Asia, Europe, and Africa, and Australia, but it's cultivated almost all over the world. This picture, this is a picture of leaves and seeds of Fenugreek, and the main compound of Fenugreek seeds are protein, fiber, gum, lipid, and some are more famous, such as galactomannan and alkaloids such as trigonelline, or amino acids such as 4-hydroxyisoleucine and diosgenin as the sarcogenin. This table shows the chemical constitution of Fenugreek, and we can see in this slide the chemical structures of 6 chemical constituents of Fenugreek. A is 4-hydroxyisoleucine, B is trigonelline, C is galactomannan, and D is the diosgenin, E and F vitexin, vitexin, and isovitexin. This is Fenugreek. As we know, Fenugreek has a lot of pharmacological effects, such as carminative, antibacterial, diabetic, and hepatoprotective. We can see this pharmacological effect of Fenugreek in this picture, and we know Fenugreek has a lot of Fenugreek had a lot of traditional medicinal uses in traditional medicinal references. For example, decreasing dysmenorrhea or postmenopausal symptoms, or it's useful for eczema, or is useful for increasing milk production, and other traditional uses. We can also see Fenugreek in keyboard of Vajay fossil that is similar to osteomyelitis. That's in Persian literature or in Yunani literature. But this study is focused on the effect of Fenugreek on osteoporosis. Research data, electronic database, including PubMed, Scopus, Cochrane, for any relevant studies about the effect of Fenugreek or metabolites on osteoporosis. Some are about some are about the effect of fenugreek on osteoporosis, some are about the metabolites such as trigonelline and diosgenin. For example, one study showed that nanoparticle containing fenugreek extracts improve trivial bond effects in rabbit. All of these studies are animal or in vitro studies. And another study showed that low intakes of taking low dosage of fenugreek seeds had a beneficial skeletal effect on in overoptimized rats. Another study showed that ethanol extract of fenugreek improved some biomechanical parameters of bone in overoptimized rats. And some studies is about dysgenin on osteoporosis. Dysgenin is a steroidal sapogenin that is found in fenugreek or Dioscorea spices. That dysgenin can improve bone biomechanical parameters of bone, and we can increase bone formation and inhibit bone resorption by some target and some pathways such as rank and rank-L pathway that's evaluated in animal and in vitro studies. What trigonelline is that is found in cafe or fenugreek seed had different and or sometimes opposite results. For example, in one study, has in rats, the study showed that trigonelline had beneficial effect on bone and is useful for bone health, but in another study, trigonelline had adverse effect on the skeletal system. So we can must consider this fact and because high dosage of trigonelline may have adverse effects of bone. In conclusion, we can say fenugreek, especially in low dosage or diosgenin, can be useful for bone health and for for management of osteoporosis, but trigonelline needs more clinical trial and more studies. And more clinical trials and more studies are needed to confirm this effectiveness and safety of fenugreek on bone health. Thank you for attention. This is my email, and I'm happy to answer any questions.
Thank you very much, Dr. Karimi, for your nice presentation. It was a valuable work. I'm afraid to say that we're not having enough time for Q&A, so I'm going to thank you again. And once more, and for the last time, I want to give the chance to Dr. Ashiru. If you're ready, we're going to have your speech, but you only have 5 minutes. So I hope the technical problems has been solved. Please start your presentation if you're ready. So meanwhile, if you're having any questions about the previous presentations, you can put it in the comment, and if you're at time, we can go through it. So I've just asked my colleague to make you the co-host so you can turn on your mic. I think now it's working. Please try to open your mic once more. You have the access, Dr. Ishu. Okay.
Okay. Good evening.
Yes, we have your voice. Could you please share your slides?
Okay. I'm trying to share this slide. Sorry. I'm trying to share this. What happened?
So if it's not working.
Oh, okay. It is working now. Are you seeing my screen now?
No, not yet.
No, not yet. Okay.
Okay. If it's okay with you, I would like to ask you to just send us your presentation, just record it and send us to the email of the symposium. Thank you very much. So we're going to go to the conclusion remarks and closing ceremony. I would like to ask Dr. Bahram Soltani to join us once more.
Well, I just want to appreciate everyone for joining us today for this fourth International Online Symposium on Meteria Medica. We learned a lot about different aspects of Fenugreek. We understood that it's a very important medicinal plant for gynecological, obstetrics, dermatologic, and hormonal problems. And there are currently a lot of evidence regarding the health benefits. So the supplements of Fenugreek in the market are actually highly valuable products which can be clinically used by physicians for the management of patients in different conditions. I hope you all enjoyed today's meeting, and actually I hope we have the chance to plan for future events with similar topics and structure. Please keep in touch by checking the symposium webpage. We will announce our future programs on Tehran University of Medical Sciences, Deputy of International Affairs website. So just wish you all have a good day. Some of you maybe night, and generally a good week ahead. And thank you again. Thank you very much.
Thank you, everyone, and goodbye.